In this part, I want to talk about the alveolar and systemic gas exchange. In this picture, you can see the pulmonary circulation and you can see the systemic circulation. The pulmonary circulation can deliver blood to and from the lungs, and the systemic circulation delivers blood to and from the systemic cells. In this figure, you can see the alveolar gas exchange. And it happens when we have gas exchange between the alveoli and the blood vessels which we have in our lung, pulmonary blood vessels. Here in this picture, you see that the pressure of oxygen in the alveoli is 104 millimeter of mercury, and the pressure of oxygen in the blood vessel is 40 millimeter of mercury. So the oxygen diffuses from the alveoli to our blood vessels until the concentration of oxygen on both sides become constant. And we have the opposite direction for our carbon dioxide. You see that the pressure of carbon dioxide in the blood vessel is 45 millimeter of mercury, and this pressure is 40 millimeter of mercury in the alveoli. So the carbon dioxide can go back to the alveoli from the blood vessels. This is the meaning of external respiration. And internal respiration or systemic gas exchange is here. Here you see that the pressure of oxygen in your blood vessels is 95 millimeter of mercury, and this pressure in the cells is 40 millimeter of mercury. So the oxygen can come from the blood to our systemic cells, and the carbon dioxide can go to the opposite direction because the pressure of carbon dioxide in our cell is 45 millimeter of mercury, and this pressure in our blood is 40 millimeter of mercury. So the carbon dioxide go to the opposite direction until the pressure of both of gases in our cells and in our blood vessels become equal. This is the meaning of internal respiration, when we have exchange of gases between our cells and the blood vessels. Some factors can affect the alveolar gas exchange. One of them is the anatomical feature of the membrane, the respiratory membrane. It's very important for efficiency of gas exchange. When we have large surface area, we have more exchange. When we have minimal thickness, we have more exchange. And the normal and the good surface area is 70 square meter, and a good thickness, the minimal thickness is 0.5 micrometer. And when we have these two factors, then our gas exchange can be the best. And we have some physiologic adjustment, which we call it ventilation-perfusion coupling. This is the ability of our bronchioles and the arterioles to do regulation. Ventilation change by bronchodilation and bronchoconstriction, and perfusion change by pulmonary arterial dilation and constriction. And you can see it in this picture. In this picture, you can see ventilation-perfusion coupling. Look at here. The increased pressure of carbon dioxide in air within the bronchiole can change the bronchoconstriction to bronchodilation. And when we have decreased carbon dioxide pressure in the air within the bronchiole, then the bronchodilation change to bronchoconstriction. And we have some changes in the perfusion of our blood vessels. Increased pressure of carbon dioxide or decreased pressure of carbon dioxide in our blood can change the diameter of our arteriole. Here you can see vasodilation. Vasodilation take place when we have decrease in the pressure of carbon dioxide or increase in the pressure of oxygen in our blood. The oxygen diffuses out of systemic capillaries to enter our systemic cell. This is the meaning of internal respiration. And I told you that the partial pressure gradient can do this. The pressure of oxygen in our cell is less than the pressure of oxygen in systemic capillaries, so the oxygen can go to the cells. And it continues until the pressure becomes equal on both sides. Here, I think I have a picture of that. Here, you see that the cell is more than the systemic cell, so we have the direction toward the systemic cell, and the pressure of carbon dioxide in the systemic cell is more than the blood, so the carbon dioxide go to the opposite direction until the concentration of both gases become equal. The alveolar gas exchange decreases the blood pressure of carbon dioxide, and the systemic gas exchange increases the pressure of carbon dioxide. The alveolar gas exchange can increase the pressure of blood, and the systemic gas exchange can decrease the pressure of oxygen in our blood. The next part is the transport of oxygen. This is the blood's ability for transporting of oxygen, and it depends on some factors. One of them is the solubility coefficient of oxygen. This is very low, and so very little of oxygen can dissolve in our blood plasma. But due to presence of hemoglobin in our red blood cells, and due to the affinity and tendency of iron to attract the hemoglobin, about 98% of oxygen in blood can go to erythrocyte and attach to hemoglobin. When we have oxyhemoglobin, it means that the oxygen and hemoglobin are connected together, and the oxyhemoglobin happen when they detach from each other, when they separate from each other. And carbon dioxide transport. The carbon dioxide has 3 different means of transport. About 7% of carbon dioxide can be solved in our plasma. 23% of carbon dioxide can attach to the amino group of our hemoglobin. Amino group is a part of globin part. So you need to know that the oxygen is connected to iron, and the carbon dioxide is connected to amine group of globin. And it can make carbaminohemoglobin, but the major amount of carbon dioxide dissolved in the plasma. And I talked about this reaction, and I will show you the reaction in the next picture. Look at here. In this picture, you can see how the carbon dioxide can change to bicarbonate and opposite. In this picture, you see that. Look at here. The carbon dioxide which we have in our blood can diffuse and go to our erythrocyte. When it comes to the erythrocyte, carbon dioxide and water inside the erythrocyte combine together, and by the aid of the enzyme, which we call it carbonic anhydrase, this enzyme is very important in our red blood cell, it can change to carbonic acid. Carbonic acid is very unstable. It dissociates immediately, and it can make hydrogen ion and carbonate. And the carbonate can go out of our red blood cell and come to our blood plasma. Due to the homeostasis of charges in our blood, when one negative ion go out of the red blood cell, one negative ion should come inside. So when the carbonate come out, then the chloride ion goes back to our erythrocyte. So you see that how carbon dioxide can be carried in our blood plasma. It can be carried as the carbonate ion. And the hydrogen ion, which is created here, can attach to hemoglobin and can be carried by our hemoglobin. Now look at the second picture. In the pulmonary capillaries, the reaction is opposite. This conversion is opposite. When the deoxygenated blood, when the blood full of carbon dioxide come to our pulmonary trunk and come to our lungs, we have this conversion. You remember that we have carbonate ion in our plasma. It comes to the red blood cell in the pulmonary capillary. In the red blood cell, the carbonate and the hydrogen, which is detached from hemoglobin, can bind again together and they can make and recombine again and make carbonic acid. This carbonic acid, with the aid of carbonic anhydrase, can make water and carbon dioxide. And carbon dioxide can easily diffuse from the erythrocyte and come to the blood. And then from the blood, because their pressure is more in the blood than in the alveoli, it can easily do diffusion and go to our alveoli. This is how our carbon dioxide transport in our blood, systemic capillaries and pulmonary capillaries. And the hemoglobin can transport different molecules. One of them is the oxygen. The oxygen can attach to iron. One of them is carbon dioxide, which is attached to globin part, and the other one is the hydrogen ion, which is the result of detachment, the result of a splitting of bicarbonic acid. It can attach to the globin part. Binding of one substance causes a change in the shape of the hemoglobin molecule, and it can influence the hemoglobin to bind or release the other two substances. So it's very important to know. Now we want to see how this hemoglobin can carry oxygen and what is the meaning of oxygen hemoglobin saturation curve. I hope you remember that each hemoglobin molecule contains 4 iron and each iron can carry 1 oxygen molecule. So each hemoglobin can carry 4 oxygen molecules at the same time for us. The oxygen saturation is very important for us. Look at this graph. In this graph, you see that when the pressure of oxygen increases, the affinity of oxygen and hemoglobin goes up. They can attach together better and better until we reach a part when all of our hemoglobin are attached to oxygen and we don't have any hemoglobin extra to make attachment with the oxygen. In this part, we reach the saturation. And this saturation happens in our pulmonary capillaries when we have moving oxygen from our alveoli to our blood capillaries. Some factors can affect this attachment, and some factors can affect detachment. The factor which can affect detachment of hemoglobin and oxygen are very important for us because when the blood comes to our tissues in systemic circulation, the hemoglobin should release oxygen and the oxygen should go to our cells and use in our cells. The factors which can help this detachment is one of them is temperature. Elevated temperature diminish hemoglobin's hold on oxygen. It means that when the temperature goes up, separation of hemoglobin and oxygen become easier. And it happens when your metabolism increase. At the time of increased metabolism, you can make more heat, and this heat helps this separation. So the oxygen and hemoglobin are separated from each other, and oxygen can go to your cell easily and used by your cell when your cell wants to make ATP, aerobic ATP production. The other factor which can affect this affinity and tendency is hydrogen, which is bound to hemoglobin. You know that when the amount of carbon dioxide in your blood increase, the amount of hydrogen ion goes up and hydrogen attached to our hemoglobin molecule. When we have combination of hemoglobin and hydrogen, the hemoglobin doesn't like to attach to oxygen that much, so they can release each other. And we call it Bohr effect. So when you have more carbon dioxide and when you have more hydrogen as the result, hydrogen ion bind to hemoglobin, it makes some conformational changes, and the tendency and affinity of oxygen and hemoglobin come down. The other factor which can affect this tendency is the presence of one molecule in our erythrocyte, which we call it 2,3-BPG. BPG is the abbreviation of biphosphoglycerate. Biphosphoglycerate is the product in our cells when our cells are very busy for making ATP. So when your cell is very busy to make ATP, it means that your cell needs more oxygen. So when you have more BPG, the tendency and the affinity of oxygen and hemoglobin decrease. Oxygen separate from hemoglobin, go to your cell to make more ATP in your cell. Some hormones can increase the amount of BPG in our erythrocyte, like thyroid hormone, epinephrine, growth hormone, and testosterone. The other one is the amount of carbon dioxide. When we have more carbon dioxide in our blood, it binds to the hemoglobin. It makes some changes in our hemoglobin. So separation of oxygen and hemoglobin become more. The factors which decrease oxygen affinity to hemoglobin is called shift right, like increased temperature, increased hydrogen ion, increased pressure of carbon dioxide. And when the affinity of oxygen and hemoglobin increase, we call it shift left, like decreased temperature, decreased hydrogen ion, and decrease the amount of carbon dioxide in our blood. In this picture, you can see the oxygen and hemoglobin and how they work together. In the first picture, you see that the most important variable which influence oxygen release from our hemoglobin during systemic gas exchange is the pressure of oxygen in our blood. So, when the pressure of oxygen in our blood is more than the pressure of oxygen in our cell, then the oxygen and hemoglobin detach from each other and oxygen move to our cell. Other variables which can change this tendency and affinity are listed here. Increased temperature. When the temperature goes up, the affinity of oxygen and hemoglobin decrease and the oxygen can go to our cell. This increased temperature happens during metabolism. The other one is the increase in the amount of hydrogen ion, which we call it Bohr effect. When the amount of hydrogen ion in our erythrocyte increase, hydrogen and hemoglobin attach together, so hemoglobin can release more oxygen. The other one is the amount of BPG. When the amount of BPG increase, BPG can attached to our hemoglobin, and BPG can push our hemoglobin to release more oxygen. And the other one is the amount of carbon dioxide. When the amount of carbon dioxide increase in our blood, it attached to hemoglobin, so hemoglobin can release more oxygen. These are the factors which can affect the tendency of oxygen and hemoglobin. In this picture, you can see the summary of respiration. Look at here. The first part in inhalation or in the respiration is called pulmonary ventilation or inspiration. The respiratory centers stimulate respiratory muscles to go to contraction. Pleural cavity and thoracic cavity volume increase, and intrapleural and intrapulmonary pressure come down, so the air can come inside our lungs. After that, we have alveolar gas exchange or external respiration. In this step, the pressure of oxygen in our alveoli is more than the pressure of oxygen in our blood, so the oxygen do diffusion and come to our blood. Then the oxygen should be transported in our blood. How it can be transported? By attaching to the iron part of hemoglobin. About 98% of oxygen is carried by hemoglobin, and less than 2% is dissolved in our plasma. And we talked about the factors which can affect the tendency and affinity of hemoglobin and oxygen. Increased temperature can decrease this affinity, so more oxygen is separated. Increased hydrogen ion, increased biphosphoglycerate, and increase the amount of carbon dioxide. All of them can increase the release of oxygen from hemoglobin. And after that, in the next picture, we have the process of expiration. The carbon dioxide which is produced from our cell do diffusion and come to our blood flow. About 7% is dissolved in our plasma, about 23% can be carried by hemoglobin, the globin part, and about 70% is transported by carbonate ion. Then this transportation take place, and by the pulmonary capillary it comes near the alveoli, and in the alveoli, because the pressure of carbon dioxide in our blood is more than the alveoli, we have the reverse and the opposite diffusion. The carbon dioxide go to our alveoli, and at that time the respiratory center decrease the stimulation of respiratory muscles, pleural cavity and thoracic cavity volume decrease, and the intrapleural and intrapulmonary pressure increase, and the air can go out. This is the process which we have at the time of expiration. We have a condition which we call it physiologic jaundice, and it's about the fetal hemoglobin. You need to know that the fetal hemoglobin in the body of fetus is different from the hemoglobin of mother or maternal hemoglobin. The fetal hemoglobin has a greater affinity to bind to oxygen when we compare it with adult hemoglobin. So, at the site of placenta, more oxygen can move from the blood of mother to the blood of fetus. After birth, this extra hemoglobin should go out, and when they are broken down, some parts of this hemoglobin can go to the skin and make the skin become yellowish color. And this is the cause of the physiologic jaundice. And when we use UV radiation, we help breaking down of these pigments, so these pigments can go out of the body easily. Now you can answer these questions. Why is so little oxygen dissolved in the plasma? How is most carbon dioxide transported in the blood? What does increased temperature do to hemoglobin's hold on oxygen? And what sort of shift to the saturation curve is caused by factors that decrease the affinity between oxygen and hemoglobin? The other part which I want to talk about is the hyperventilation and hypoventilation on the function of our cardiovascular system. The hyperventilation is a breathing rate or depth that is increased above the body's demand. Hyperventilation may be caused by anxiety, panic, or ascending to the high altitude. Hyperventilation may also be performed voluntary when you consciously inspire and expire excessively at the accelerated rate. Pressure of oxygen increase and the pressure of carbon dioxide decrease in our alveoli. When the pressure of carbon dioxide comes down, we call it hypocapnia. The low blood carbon dioxide pressure can make vasoconstriction, and one part which we have vasoconstriction is the blood vessels in our brain. The blood vessel in our brain go to vasoconstriction, and it can decrease the amount of oxygen in our brain, and it can make faint, dizziness, numbness, tingling, cramp, and tetany, all of them. At this time, the amount of hydrogen in our blood decrease. And when the amount of hydrogen in the blood comes down, we call it respiratory alkalosis, because the blood becomes basic in its pH. And if the hyperventilation prolonged, it can make disorientation, loss of consciousness, coma, and possible death. The meaning of hypoventilation is when you breathe too slow. We call it bradypnea. Bradypnea is the other name. And sometimes the breathing pattern is too shallow, which we call it hypopnea. And the causes which can make hypoventilation are the obstruction of airways, pneumonia, the injuries in our brain stem, and also respiratory conditions. At this time, the level of oxygen is down and the level of carbon dioxide goes up. When the level of oxygen comes down, we call it hypoxemia, and it can lead to low oxygen in the tissue, which we call it hypoxia. And at this time, the level of our carbon dioxide increase, and the increase in the level of carbon dioxide is called hypercapnia. When we have hypoventilation, we don't have enough oxygen delivery, and it results in increase in the concentration of hydrogen ion due to increase in the level of carbon dioxide. When the level of hydrogen ion goes up, we have a condition which we call it respiratory acidosis. And it may cause lethargy, sleepiness, headache, polycythemia, and the cyanotic tissue. The tissue become black in their color because they don't have enough oxygen. And if it's prolonged, convulsion, a loss of consciousness, and death can occur. So the pH of our blood is very important. And the respiratory system is one of the systems which can do regulation of this pH. Besides the respiratory system, we have urinary system which work with the pH of our blood, and I will talk about it later for you. And breathing and exercise are affected each other. While we are doing exercise, breathing shows hyperapnea to meet increased tissue need. At the time of exercise, your tissue needs more oxygen, so you need to do hyperventilation. And blood pressure of oxygen and blood pressure of carbon dioxide should remain constant. At the time of exercise, the cellular respiration increase. The cardiac output goes up and the blood pressure increase. And all of them can help the homeostasis of oxygen and carbon dioxide pressure in our blood. And the respiratory center is stimulated from one or more causes. You know that the proprioceptors can send information when you are doing exercise, and the motor output from the cerebral cortex can send information to our respiratory system, and conscious anticipation of exercise. We do more breathing, and we can make homeostasis and keep the pressure of oxygen and carbon dioxide in constant level. And now you can answer this question. If hyperventilation is excessive, what change occur in the blood pH? And what distinguishes hyperventilation from hyperapnea? Here we are done with the respiratory physiology. Let me know if you have any questions in this part.